In this video, we're going to look at the process of protein synthesis. Protein synthesis can involve the production of chemical proteins, also biological proteins like enzymes. So let's have a look and see how these proteins are made. Okay, so here is our cell. Inside the cell is the cell nucleus. The cell nucleus contains our two strands of DNA. This section of DNA here is going to code for our protein, so it contains all the information required to assemble the correct protein. Hopefully we can remember that proteins are made up of different amino acids. The code in the DNA is going to tell the ribosome, the organelle that makes the protein, what order to put those amino acids in to produce the correct protein. Outside of the cell nucleus in the cytoplasm is the ribosome. As I said, this is the organelle, the part of the cell that is going to produce our protein. So the first stage in the production of a protein is the DNA strand in the nucleus splits apart. It unzips. And we then have a new copy of the DNA made, and that's done onto a strand of mRNA. The mRNA stands for messenger RNA. So a new strand is added, a new backbone, and complementary bases are added. You will notice that the base T, which normally pairs with A, is replaced with the base U. So G's pair with C's, T's pair with A's. A's would normally pair with the T, but instead we've got a U replacing it. Once our mRNA strand has been assembled, that can then leave the cell nucleus. So we're not taking the whole strand of DNA, we're taking the section of relevant DNA, copying it onto an mRNA strand, and that mRNA strand is then leaving the cell nucleus. Okay, and it's going to go to the ribosome. Once the mRNA is in the ribosome can read sequences of bases, and it's going to look at 3 bases in a row. This is called a triplet. Once it's read those bases, each set of 3 bases has a corresponding amino acid that gets added. So our ribosome reads the first 3, and the correct amino acid is then added. It reads the next 3 bases, so UUG in this case, and a different amino acid is added. The next 3 bases, the third amino acid is added, and so on, until the full protein has been assembled. As these amino acids are read on the codons, the triplets, they are then joined together as well. And the joining together of amino acids forms our protein. So to summarise, we'll have a look at the key stages and the key pieces of information you need to understand about protein synthesis. The first piece of information is to understand what a protein is. A protein is a sequence of amino acids that are all joined together. Different proteins have a different order of amino acids. The information to create that sequence of amino acids and the order to put them in is stored inside the DNA. So to create your protein, you need to get the information out of the DNA and to the relevant part of the cell. A strand of DNA is quite large and is unable to exit the cell nucleus easily. So the relevant information, the relevant strand of DNA, is copied onto an mRNA, a messenger RNA strand, which can quite easily leave the cell nucleus because it's sort of smaller than a strand of DNA. So the mRNA strand travels to the ribosome. Once it's at the ribosome reads the sequence of bases and then assembles the amino acids into the correct order to produce the protein. Those amino acids are joined together, you then have your completed protein chain.